In this lecture, I'm going to talk to you more about the connectivity map, or the CMAP, that we discussed in the previous lecture as a method to validate expression to kinases. So to remind you, the original connectivity map was a dataset published by members of the Broad Institute who performed over 6,000 microarray experiments where they stimulated 4 human cancer cell lines with FDA-approved drugs applied at different concentrations. The gene expression changes were measured after 6 hours. So for each perturbation, we can obtain a list of up and down regulated genes. On the CMAP website, you can query this dataset to find drugs that overlap with the user input lists of the up and down gene. And this can be used to find drugs that can potentially reverse the expression of cells in a desired direction. For example, if your input list from a disease versus normal cells, or if you want to identify drugs that have a similar effect to the observation that you are obtaining in your own experiments, where you may be measuring the effects of novel drugs, or if you knock down specific genes and those knockdowns induce changes in expression that can potentially be mimicked by drugs. So we came up with a simple method to search for pairs of drugs instead of searching for single drugs, as it is possible with the connectivity map website. So what we've done, we first look for the 500 up genes and 500 down genes from CMAP for each perturbation, and this results in 2 gene set libraries, one for the up genes and one for the down genes. Then, given an input of up and down genes from the user, we count for each entry how many genes every pair of drugs can reverse maximally, downregulate the up genes, and upregulate the down genes. And then we score each possible pair for this ability to reverse the expression of the user input lists. We also check if the drugs do not aggravate the genes that are already up and down in the disease. This simple formula is shown here, and there is a freely available desktop software written in Java that can perform this analysis, and you can get it from the URL on this site. So if you remember from the previous lecture, our lab is collaborating with Zhang, John He, who is studying the kidney disease called HIVAN. So we have gene expression microarray data, TG26 mice, which are a transgenic mouse model that develops kidney disease similar to the disease observed in humans. We also have gene expression from wild-type mice with healthy kidneys. Using expression to kinases, I showed you how we identified a kinase that potentially is involved in mediating the changes that lead to the disease in these mice. So we also tried DrugPairSeeker to identify pairs of drugs that can reverse the expression changes towards the healthy phenotype. The results of this study were published recently in the journal JSON. Interestingly, the program identified captopril as a potential drug in this list of drugs, and captopril is already used to treat those patients, but a combination of captopril with an HDAC inhibitor was not tested before. I asked Zhang to further explain to you how he validated the prediction of this drug combination, and Zhang is going to talk to you a little bit next.
So my name is John He. I'm a clinical nephrologist at Mount Sinai Hospital. I'm also a professor of medicine at the Mount Sinai School of Medicine. I'm also a physician scientist, and I've been interested to look at the drug therapy for kidney disease. We don't have a lot of options to have a drug to treat patients with kidney disease. So therefore, it's very important for us to develop a therapy regimen for treating the patient with kidney disease. So in this study, collaborating with Dr. Maya, we developed this method to predict the drug combination which can provide more effective therapy for patients with kidney disease. So using this computational program, we predict the two drugs. One is the angiotensin 2 inhibitor, the other one is HDAC inhibitor. So to validate this computational prediction, we use animal model, which is a HIV transgenic mice. We also call it the TG26 mice. These mice develop a significant glomerular and tubular interstitial disease. They also develop proteinuria, really mimicking the human HIV-associated disease. Therefore, this is a great model to use actually to test the drug therapy. We use this animal model to test whether the combination of these two drugs could provide additional benefit as compared to the mice treated with one drug, or we call the monotherapy. So we essentially treat mice with either S inhibitor or HDAC inhibitor or both drugs together. And the results show that with combination therapy, the combination therapy could provide additional benefits as compared to the mice treated with either S inhibitor or HDAC inhibitor alone. We look at the kidney function. We also look at the gene expression, and we found that the combination therapy can reduce significant proteinuria as well as improve the renal function and also improve the kidney injury based on the kidney histology. Therefore, our study essentially validates our computational prediction, and we believe that the method we developed to predict the drug combination is valid and it could be applied to study kidney disease or other disease model.
So a few lectures ago, I demonstrate this canvas enrichment analysis approach. So you can see from the results that both drugs, the HDAC inhibitor and the ACE inhibitor, are both anti-inflammatory. Both are targeting the immune system of the cells. However, they appear to work through different pathways. So in general, I believe that the connectivity map idea is very exciting for systems biology as well as systems pharmacology. And this is because we can now use signature-based approach to compare the effects of drugs and identify similarity between drugs using a new dimension, which is gene expression. And the NIH Office of the Director seems to agree with me because they are investing enormous amount of resources to further develop this idea. And this is for a project called LINKS. They have provided large funds to create a new version of the connectivity map, and this is the project called LINKS. So what is LINKS? LINKS stands for Library of Integrated Network-Based Cellular Signatures. It aims to catalog a large amount of gene and protein expression profiles after a variety of perturbing agents applied to different human cells. And this large-scale expression data collection can be used to obtain a systematic global understanding of the cellular response to cellular perturbation and completely map the landscape of the possibility of a cell to respond to various perturbations. So most of the LINKS data is generated through the L1000 technology. The L1000 measure 1,000 mRNAs in each experiment, and the rest of the transcriptome is estimated by a model built from a large set of microarray data from the Gene Expression Omnibus. The L1000 technology is cost-effective, costing only $2 or $3 per experiment, and it is suitable for genome-wide expression measurements in high throughput. So far, the researchers at the Broad performed almost 1 million of those experiments. The dataset is organized in 3 levels. You can get the raw measurements, normalized data, or the Z-score signatures. And this is going to be available very soon to the public. So Chanan Duan, who is a PhD student in the lab, who you will meet later in the course, where he will present methods to perform principal component analysis and hierarchical clustering, developed 2 web applications to query and browse the new version of the connectivity map. So let me demonstrate to you the tool online.
This browser was developed to visualize and analyze L1000 experiments. There are 3 batches of those experiments. Each is about 3,000 experiments measured using the L1000 technology. These batches are called LGP001, LGP002, and LGP004. LGP001 and 02 are drug perturbations. Each of them have about 80 drug, which is mostly kinase inhibitors, and LGP004 are endogenous ligands applied to 6 cancer cell lines. So what we've done, we divided the results into the 3 batches, and also for each cell line we have a different canvas. So these red canvases that you can see here are representing all the perturbations for a specific cell line in a specific batch. The red view, or the first view, the default view, is called the average change view, and it shows how much each of the perturbations affected the overall changes in expression in those cells in this particular batch. You can use this slider to increase or decrease the opacity, and this will help you to identify the perturbations that were the most significant, the drugs or the ligands that actually made a significant change into the expression of those cells. You can also look at clustering by time point. You can have a view of all of the drugs or ligands that were used and the dose. One of the useful things about the drugs view is that it helps you to locate specific drugs of interest. So for example, if I select a specific drug, I can see the perturbations where the drug is. The location of the perturbations for a specific cell line and specific batch is the same whether you're looking at average change, time point, or drug. So for HERG, in general, there was not a significant perturbation, maybe a little bit in these 2. At 6 hours with 100 nanomolars or 1 nanomolar. The most significant perturbations in this particular grid for MCF7 cells was with this ligand MS at 6 hours, 1 nanomolar. If I change the cell type and identify another significant perturbation, I can look at this androgen receptor AR, 24 hours, 100 nanomolars. And what I've done also, I clicked on this particular experiment or particular condition. So this is actually a merge of 4 repeats of the same condition were merged, and the average change was recorded. So when you click on the actual experiment, you can see enrichment analysis. For example, here it's KEGG pathways. And what you're looking at on the right, those are gene set libraries, also organized on a canvas, where each square represents a pathway or a gene set. And the pathways are organized based on their similarity on this grid. And when you click, the view changes to this p-value view, where the most significant pathways, the most overlapping pathways, are marked in bright colors, and the rest of the pathways that don't have any overlap. So besides KEGG pathways, you can try different things. For example, very popular is the gene ontology analysis. So if I click on another significant perturbation, for example this one, I can get the enriched Go terms, and you can also see it in a table. So this is the regulation of cytoskeleton organization, and you can also see it in the p-value view. This is one of those bright spots. I can also look at the downregulated genes, and these are different terms for gas 6 hours, 1 nanomolar. If I look at the table, I can see that there is the downregulation of S phase of the mitotic cell cycle. So this is basically the features of this Canvas Browser 1. I hope you find this demo useful, and if you have any suggestions, please contact us. We are always happy to make improvements. And also there is another Canvas Browser 2 that you can check. It has very similar features to this first canvas, but on the right you populate gene lists, and then the analysis is done by an external tool called Enricher.
So finally, I would like to discuss with you this idea of looking at drug perturbation in expression space. So once you listen to the principal component analysis lectures by Neil Clark and Chana and Duan, you will see how we can map expression vectors into a lower dimensional space, a principal component space, and this places the expression vectors in each cell under each condition in this three-dimensional space, and we can now navigate this space by trying to push cells in various directions using various types of perturbations. So here you see a small example of the various possible perturbations from some data processing that was done by Chaunan and Neil, and you can see how gene expression is changing from the control towards those various directions by this variety of treatment. Various ligands and drugs push cells to various directions, so potentially we can design drug combinations and understand this landscape of where cell goes after various perturbations.